On January 10, 2025, researchers from the Broad Institute of MIT and Harvard alongside Cyrus Biotechnology announced a groundbreaking advancement in CRISPR technology, the engineering of CRISPR enzymes that can evade the human immune system. This innovative development aims to enhance the safety and effectiveness of gene therapies, a vital consideration as CRISPR technologies are increasingly utilized in clinical settings. Background of the study. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, represents a revolutionary tool in gene editing widely used in various fields including gene therapy and cancer research. However, a significant challenge arises from the fact that about 80% of the population has pre-existing immunity against the bacterial nucleases used in CRISPR, primarily due to exposure to these bacteria in everyday life. This immune response can lead to reduced efficacy of CRISPR therapies and increased side effects, underscoring the need for enhanced safety measures. Discovery process. The research team led by Professor Feng Zhang utilized advanced mass spectrometry to identify specific protein sequences within the Cas9 and Cas12 nucleases that trigger immune responses. The investigation revealed 3 short sequences, approximately 8 amino acids long, in both Cas9 from Streptococcus pyogenes and Cas12 from Staphylococcus aureus that were recognized by the immune system. To address this issue, the team collaborated with Cyrus Biotechnology, employing structure-based computational tools to design new versions of these nucleases that lack the immunogenic sequences while retaining their gene editing functionalities. This strategy was bolstered by computational modeling to predict the likelihood of immune recognition of the engineered nucleases. Key findings. 1. Immune evasion. The engineered Cas9 and Cas12 nucleases were shown to evade immune detection, demonstrating similar gene editing efficiency compared to their traditional counterparts while significantly reducing immune responses in mouse models. 2. Innovative methodology. The research combined AI-driven protein design with empirical laboratory data to create more effective and safer nucleases. This interdisciplinary approach proved crucial in addressing the challenges posed by immune responses to CRISPR therapies. 3. Clinical implications. The findings were published in Nature Communications and suggest that this engineering could lead to safer and more effective gene therapies. Professor Zhang emphasized the growing need for safety as CRISPR therapies transition into clinical applications, noting that further research could also focus on designing immune-evading delivery vehicles for these therapies. Future prospects. The implications of this research extend beyond just CRISPR enzymes. The methods developed could pave the way for the design of various therapeutic tools that can evade the immune system, potentially revolutionizing how gene therapies are administered. The team plans to explore how these engineered nucleases can be utilized in different therapeutic contexts, aiming for broader applications in treating genetic diseases and cancers. Timeline of CRISPR development. The journey of CRISPR technology has been marked by significant milestones. 2013, the Zhang Lab published the first method to engineer CRISPR for genome editing in mouse and human cells. 2025, the latest research detailing the immune-evading CRISPR enzymes was published, highlighting a significant advancement in the field. Conclusion. The ability to engineer CRISPR enzymes that evade the immune system marks a significant step forward in the quest for safer and more effective gene therapies. By addressing the critical issue of immune response, this research not only enhances the potential of CRISPR technology but also opens new avenues for clinical applications. As scientists continue to refine these techniques, the future of gene therapy looks increasingly promising, with the potential to treat a range of genetic disorders more safely and effectively. The recent advancements in CRISPR technology, specifically the engineering of CRISPR enzymes that can evade the immune system, introduce several counterintuitive insights worth noting. 1. Immunogenicity of bacterial proteins. A primary finding of the research is that CRISPR enzymes, which are derived from bacteria, can trigger immune responses in humans. This is counterintuitive because one might assume that these proteins being naturally occurring in bacteria would be benign to the human immune system. However, the study reveals that about 80% of people have pre-existing immunity to these enzymes due to prior exposure, which can undermine the efficacy of CRISPR-based therapies. 2. Designing immunogenic sequences out. Traditionally, one might think that the effectiveness of CRISPR enzymes is primarily dependent on their ability to edit genes rather than their interaction with the immune system. The research challenges this notion by demonstrating that modifying specific short protein sequences to eliminate immunogenicity can enhance the overall effectiveness of gene therapies. This highlights the importance of considering the immune response as a critical factor in the design of gene editing tools. 3. AI in protein design. The use of AI-driven protein design in this study leads to the creation of new Cas9 and Cas12 enzymes that evade immune recognition. This is counterintuitive in the sense that one might not expect computational models to significantly outperform traditional experimental methods in biological contexts. The success of AI here suggests a paradigm shift in how biological engineering can leverage computational tools to solve complex biological problems. For maintaining function while reducing immune recognition, the engineered CRISPR enzymes retain similar gene editing efficiencies compared to their traditional counterparts, yet elicit much lower immune responses. This finding is counterintuitive because it implies that reducing the recognition of the enzyme by the immune system does not come at the cost of effectiveness, which one might typically expect in biological systems where trade-offs are common. 5. Broader implications for gene therapy. The study emphasizes that the advancements in minimizing immunogenicity are just one piece of the puzzle for safer gene therapies. This is counterintuitive because while the focus is on CRISPR enzymes, the broader implication is that all aspects of gene therapy, including delivery methods and other components, must also be considered in conjunction with the immune response. It suggests a more holistic approach to designing gene therapies may be necessary, rather than a narrow focus solely on the editing mechanism. 6. Future research directions. The researchers anticipate that designs for immune-evading delivery vehicles could be developed using similar principles. This is counterintuitive because one might expect that the focus would remain exclusively on the enzymes themselves. Instead, it reflects an understanding that the entire therapeutic strategy, including how therapies are administered, plays a crucial role in their success. In summary, the engineering of CRISPR enzymes that evade the immune system reveals unexpected relationships between immunogenicity and therapeutic efficacy, the potential of AI in biological design, and the necessity of a comprehensive approach to gene therapy development. The recent advancement in CRISPR technology, particularly the engineering of CRISPR enzymes that can evade the immune system, is a significant breakthrough with profound implications for gene therapy. While many people may focus on the basic premise of improving gene editing efficiency, several crucial aspects of the research's implications, methodology, and potential future directions are often overlooked. Key content overlooked. 1. Immune response and gene therapy. A critical element of this research is the focus on the immune response to CRISPR nucleases. Most discussions around CRISPR technology concentrate on its editing capabilities, but the fact that approximately 80% of the population has pre-existing immunity to these bacterial-derived enzymes significantly hampers their therapeutic potential. This immune response can lead to reduced effectiveness of treatments and increased side effects. By engineering nucleases that evade this immune recognition, the research provides a solution to a major barrier in the clinical application of CRISPR technologies. 2. Identification of immunogenic sequences. The study utilized advanced mass spectrometry and computational modeling to identify specific protein sequences within the Cas9 and Cas12 enzymes that trigger immune responses. This meticulous identification process is significant because it not only informs the design of new enzymes but also enhances our understanding of how the immune system interacts with foreign proteins. This knowledge can be leveraged in future studies, potentially broadening the scope of CRISPR applications beyond gene therapy to include other therapeutic areas where immune reactions pose challenges. 3. Combination of AI and experimental techniques. The research exemplifies a successful integration of artificial intelligence and traditional experimental approaches in protein engineering. By using AI-driven protein design alongside laboratory data, the researchers were able to create more effective and safer CRISPR nucleases. This methodological innovation could set a precedent for future biotechnological research, emphasizing the role of computational tools in overcoming biological challenges. This intersection of AI and biology could lead to rapid advancements in various fields, including synthetic biology, vaccine development, and personalized medicine. 4. Potential impact on delivery systems. The researchers hinted at future work on designing delivery vehicles that also evade the immune system. This aspect is particularly significant as the efficacy of gene therapies often hinges on the delivery mechanism. Current delivery systems can themselves provoke immune responses which can lead to treatment failures. By developing methods for immune evasion in both the editing enzymes and their delivery systems, there is potential to revolutionize how gene therapies are administered, increasing their safety and effectiveness. 5. Clinical implications. While the paper discusses the potential for safer and more efficient gene therapies, the broader implications for clinical applications are profound. As CRISPR therapies move closer to clinical use, ensuring their safety becomes paramount. This research not only addresses immediate concerns regarding immune responses but also lays the groundwork for regulatory approval processes that could facilitate the faster introduction of CRISPR-based therapies into the clinic. 6. Historical context and future perspectives. The research builds on a historical foundation laid by the Zhang Lab, which has been pivotal in the development of CRISPR technology since its inception in 2013. Recognizing the timeline of CRISPR advancements provides context to the current work and highlights the rapid pace of innovation in the field. As CRISPR technologies continue to evolve, understanding previous milestones can inform future developments, including potential expansions of CRISPR applications to address complex genetic disorders. 7. Broader applications beyond gene therapy. The implications of this research extend beyond just gene therapy. The ability to engineer proteins that evade immune responses could also influence the development of CRISPR-based diagnostic tools, vaccines, and even treatments for autoimmune diseases. This versatility is often not discussed in detail but represents a significant area of impact that could reshape various aspects of medicine and biotechnology. Significance of overlooked aspects. The significance of these overlooked aspects lies in their potential to reshape our understanding of CRISPR technology and its applications. By addressing the immune response, researchers are not just improving a tool, they are enhancing a platform that could lead to breakthroughs in treating genetic diseases, improving vaccine strategies, and developing novel therapeutics. The combination of AI and empirical research exemplifies how interdisciplinary approaches can yield innovative solutions to long-standing challenges in biology. Moreover, the emphasis on safe delivery systems is crucial for the practical application of any therapeutic modality. As the field progresses toward clinical use, these considerations will be paramount in ensuring patients' safety and therapeutic efficacy. In conclusion, the engineering of CRISPR enzymes that evade the immune system is a landmark achievement that offers deeper insights into the intersection of immunology, protein engineering, and gene therapy. The implications of this research extend far beyond immediate gene editing capabilities, with the potential to influence a wide range of biomedical applications. Recognizing and exploring these nuances will be essential as the scientific community continues to advance the frontiers of genetic medicine. 1. Immune evasion in CRISPR technology. The engineered CRISPR enzymes Cas9 and Cas12 are designed to evade the immune system, which is crucial for enhancing the safety and effectiveness of gene therapies. Approximately 80% of individuals have pre-existing immunity to these enzymes due to natural exposure, leading to potential complications in therapeutic applications. By identifying and modifying the immunogenic sequences in these enzymes, the researchers succeeded in creating variants that maintain gene editing efficiency while significantly reducing immune responses, thus addressing a major hurdle in the clinical application of CRISPR. 2. Innovative research methodology. The study utilized a combination of advanced techniques including mass spectrometry and AI-driven protein design to pinpoint the specific protein sequences responsible for triggering immune reactions. This interdisciplinary approach demonstrates how integrating computational modeling with experimental data can lead to significant advancements in biotechnology. By collaborating with Cyrus Biotechnology, the team not only identified the problematic sequences but also engineered modified nucleases that lack these immunogenic portions, showcasing a robust method for tackling complex biological challenges. 3. Implications for future gene therapies. The findings of this research hold substantial promise for the future of gene therapy as they pave the way for safer and more effective treatments. With the potential to minimize immune-related side effects, these engineered enzymes could enhance the therapeutic outcomes of CRISPR applications in various medical conditions, including genetic diseases and cancer. The researchers also hinted at future studies aimed at designing immune-evasive delivery vehicles, indicating a broader impact on how gene therapies can be administered safely and effectively. For significance in the context of CRISPR evolution, this research represents a significant evolution in CRISPR technology, which has been rapidly advancing since its inception in genome editing. The study, led by prominent figures in the field including Feng Zhang, not only contributes to the ongoing development of CRISPR methodologies but also highlights the importance of ensuring safety as these technologies transition into clinical settings. By addressing the immunogenicity of CRISPR systems, this work reinforces the need for continuous innovation in the field to realize the full potential of gene editing while safeguarding patient health. 1. The development of the polio vaccine. 1950s. In the early 1950s, Dr. Jonas Salk developed the first effective polio vaccine. Polio caused by the poliovirus was a major public health threat, leading to paralysis and death, particularly in children. Salk's vaccine was based on an inactivated form of the virus, which allowed the immune system to build defenses without causing disease. The introduction of this vaccine led to a dramatic decline in polio cases worldwide. However, as the vaccine was rolled out, concerns arose about its safety and the potential for vaccine-derived poliovirus. This resulted in ongoing research to improve vaccine formulations and delivery methods, paralleling the current efforts to engineer CRISPR enzymes to evade immune responses. 2. The HeLa cells controversy. 1951. HeLa cells derived from Henrietta Lacks without her consent became one of the most important tools in medicine. These cells were used in research for decades, contributing to the development of the polio vaccine, cancer treatments, and more. The ethical implications of using human cells without consent highlighted the need for informed consent and ethical standards in biomedical research. This historical case resonates with contemporary discussions about genetic editing and the ethical considerations surrounding the use of CRISPR technology, especially as it pertains to human health and genetic modification. 3. The gene therapy trials for SCID. 1990. In 1990, the first gene therapy trial took place targeting severe combined immunodeficiency (SCID), a genetic disorder that severely weakens the immune system. Researchers used a retroviral vector to introduce a functional copy of the gene into the patient's cells. While the initial trial showed promise, later trials encountered serious complications, including the development of leukemia in some patients due to insertional mutagenesis. This underlined the risks associated with gene therapy and the importance of ensuring safety in genetic interventions, similar to the recent advancements in engineering CRISPR enzymes to minimize immune responses in therapeutic applications. The recent engineering of CRISPR enzymes that can evade the immune system marks a significant advancement in the field of genetic editing and therapy. This breakthrough, spearheaded by researchers at the Broad Institute of MIT and Harvard in collaboration with Cyrus Biotechnology, opens up a promising future in gene therapy, particularly in terms of safety and efficacy. Future predictions. 1. Enhanced gene therapy applications. The ability of these engineered CRISPR enzymes to bypass immune detection is expected to expand the range of gene therapies. As these therapies advance, they could effectively treat genetic disorders, cancers, and other diseases previously deemed difficult to manage due to immune responses. By minimizing immune reactions, patients could receive more consistent and effective treatment regimens. 2. Clinical trials and regulatory approvals. As researchers gather more data on the efficacy and safety of these new CRISPR enzymes in animal models, we anticipate a surge in clinical trials. Regulatory agencies may see an expedited review process for these therapies, given their potential to reduce adverse immune responses. This could lead to the first human applications of these modified enzymes within the next few years. 3. Integration with AI in computational biology. The use of AI in designing proteins that evade immune recognition is likely to become a standard practice. This innovation could lead to more sophisticated models that anticipate immune responses, facilitating the development of a new generation of CRISPR tools. The convergence of AI and genetic engineering will likely enhance the precision of gene editing and reduce off-target effects. For broader therapeutic delivery systems, research may extend beyond CRISPR enzymes to include the engineering of delivery systems that can effectively transport these tools without triggering immune responses. This could involve designing nanoparticles or viral vectors that are optimized for low immunogenicity, further enhancing the efficacy of gene therapies. 5. Personalized medicine. As the field of gene therapy progresses, we may see a shift towards personalized medicine approaches where treatments are tailored to the individual's genetic makeup and immune profile. This could lead to more effective therapies for a range of diseases, improving patient outcomes and minimizing side effects. 6. Ethical considerations and public acceptance. With the advancements in CRISPR technology, ethical discussions around gene editing will intensify. Public discourse regarding the implications of genetic modifications, especially in humans, will need to evolve alongside the technology. Clear communication about the benefits, risks, and ethical considerations will be essential for public acceptance and regulatory support. 7. Global health implications. The ability to engineer non-immunogenic CRISPR enzymes could have profound implications for global health, particularly in developing countries where access to effective treatments is limited. These advancements might enable broader implementation of gene therapies for diseases prevalent in these regions, such as genetic disorders and infectious diseases. 8. Interdisciplinary collaborations. The complexity of developing safe and effective CRISPR therapies will likely foster interdisciplinary collaborations among biologists, clinicians, computational scientists, and ethicists. This collaborative approach can lead to comprehensive strategies for tackling the multifaceted challenges posed by genetic diseases. 9. Long-term monitoring and safety studies. As these therapies enter clinical settings, long-term monitoring will be critical. Researchers will need to establish robust frameworks for tracking patient outcomes and any late-onset side effects, ensuring that the therapies remain safe and effective over time. 10. Advancements in related technologies. The success of engineered CRISPR enzymes may spur advances in other gene editing technologies such as CRISPR-Cas13 and base editing. These technologies may also be refined to minimize immune responses, broadening the toolkit available for genetic engineering. Conclusion. The engineering of CRISPR enzymes that evade the immune system is poised to transform the landscape of genetic therapy. With advancements in technology, an increase in clinical applications, and a focus on safety and efficacy, the future of gene editing appears bright. As we move forward, it is essential to navigate the ethical landscapes, regulatory frameworks, and public perceptions that accompany such revolutionary changes in medicine. The next decade will likely witness an unprecedented integration of genetic engineering into personalized healthcare, fundamentally altering the treatment of genetic and acquired diseases.